You'll probably see this slide at the beginning of every talk from this conference, and so sorry to bore you. But in essence, exactly like Alex sort of was describing, AGAT activity is localized and has historically been localized to the kidney. This information evolved even half a century ago. So people took homogenates of different organs and looked for the activity of these enzymes. And so it was known kind of out of the 50s and 60s that you could homogenize a kidney and detect high levels of an enzyme that would take arginine and glycine and convert them into guanidinoacetate and ornithine. And then it was known that slices of the liver were very good at converting that guanidinoacetate into creatine, and so that GAMT activity was found to be highly localized to the liver, or at least at its highest abundance. And then once these two organs were thought to perform this synthetic pathway, the product, which is creatine, was exported to the rest of the body and used for powering the cellular ATP cycle, the phosphagen system, by being reversibly phosphorylated by creatine kinases. And in order to get into the cell, in order to do that, it had to be transported across the membrane by creatine transporter 1, which is encoded by SLC6A8. And so importantly, cell types that use a lot of creatine to function are sort of marked by having a high concentration of mitochondrial creatine kinase because that's important for initially phosphorylating creatine into phosphocreatine for then downstream consumption. So when I was a postdoc, one thing I wanted to know was, is this dogma true across the body? And so at that time, a lot of single cellular experiments were happening, and I'll show you some data in the next slide. But initially what I did was I took tissues from across the body and I stained for AGAT and GAMT. And sure enough, just like the dogma suggested, if you stain for GAMT inside the liver, you see high levels of GAMT. But importantly, it was in hepatocytes. And so we can mark the hepatocytes with this marker albumin. And in contrast, we can mark the endothelium. So the cells that line your blood vessels, we can mark them with a different marker. And when we do that, we actually were able to detect AGAT activity inside the liver, or AGAT presence inside the liver. And it was coming from the endothelium. And just to show you that in 3 dimensions, this is a mouse's liver that I've taken serial sections using a confocal microscope. And if you look at where AGAT is expressed inside the liver, you get these little spots of red. It may be hard to appreciate with the lighting, but suffice it to say that it overlaps with the endothelial cell marker. And so we thought about this project because in 2018, there was a nice publication where they took a mouse and they separated all the cell types in the mouse's body and then measured RNA expression in each cell type. And when you do this, you get a map of where each gene is expressed throughout the entire mouse body at single cell resolution. And when I plotted this map, what I found was that the gene that encodes AGAT, which is GATM, is very, very highly expressed, like you would think, inside the kidney, inside the proximal convoluted tubule cells of the kidney. But in blue is the liver, and lo and behold, it was detected at the RNA level in the endothelium of the liver, whereas the hepatocyte in the liver expresses high levels of GAMT. And so when we made this plot, one thing that stuck out to us was this big cell up here. So those are oligodendrocytes in the brain. And so even though the dogma had been that the kidney and liver participate in forming creatine for export to the rest of the body, it looked to us like the oligodendrocyte was like a powerhouse that could be generating creatine all by itself. And importantly, there was a high degree of overlap of both AGAT and GAMT expression in oligodendrocytes. So we turned again to immunofluorescence and we measured AGAT and GAMT within the brain. And here I'm showing you a section of mouse cerebellum. In green, I've highlighted the neurons. And the nice part about the cerebellum is it's very layered. So the neurons all bundle together in this granular layer here. But then what you get is in this white matter here, it's very enriched for oligodendrocytes. And that's where we're seeing the signal for AGAT. And similarly, within this clear area in the center, where you have lots of oligodendrocytes. We also saw a lot of signal for GAMT, so confirming the fact that oligodendrocytes express a lot of AGAT and GAMT. And so we came up with a model for how creatine could be produced within the brain and then potentially used. And so our model was that oligodendrocytes are known to be these nurse cells for neurons, and they allow them to myelinate their axons and transmit signals very quickly throughout the brain and the rest of the body. And so maybe these oligodendrocytes are little factories for creatine inside the brain. And then once they've stitched together creatine by expressing both AGAT and GAMT, they export it via an unknown channel to be taken up by neurons using CT1, and then enriched and then used by those neurons. And the reason we think that neurons are probably the relevant cell type for creatine inside the brain, although probably other cell types use it a little bit, is that the neurons express this mitochondrial creatine kinase. And that's very reminiscent of skeletal muscle and cardiac muscle, which are highly dependent on creatine for function. And so we developed this approach for gene therapy based on these data. So we hypothesized that patients with CTD have a decreased intracerebral creatine content due to a failure of neurons to uptake creatine from their neighboring oligodendrocytes. And so what we proposed to do was bypass the function of the channel by directly expressing AGAT and GAMT within neurons so that you don't need to transport it across the cell membrane. And so as a proof of concept, we started out with HEC293 T cells, which are a human epithelial kidney line. But in many ways, these cells are reminiscent of neurons. And at the gene expression level, they correlate best with cells that are from the adrenal medulla. And so they're sort of like a neuronal cell line that originated from an embryonic kidney. And so we overexpressed, so we developed lentiviral vectors to overexpress AGAT and GAMT, and then we transfected those into HEC293 T cells. And when we do this, what you can see is that when you would use an empty vector, HEC293 T cells expressed a little bit of GAMT, but when you overexpress GAMT from this lentiviral vector, you can massively upregulate the expression of GAMT. And the same was true for AGAT. And when you put them together, they didn't affect each other. So they are both expressed at very high levels. And we tagged both of these proteins with the same peptide tag so that we could use the same antibody by Western blot to detect their relative levels. And when we did that, their relative levels were about similar, maybe a little bit less for AGAT than GAMT. And then we measured, okay, how much creatine is inside of these cells when we do this. And so what we found was that you do get a boost of about something like twofold when you overexpress AGAT by itself, probably because there's a little bit of GAMT expression inside of HEC cells alone. But when you co-express both AGAT and GAMT, you can really dramatically increase the amount of creatine inside the cells by about 8-fold. So an 800% increase in the amount of intracellular creatine. To our eye, either by loading the Western blot, so using GAPDH or a marker for the nucleus, histone H3, or by measuring the total cellular protein, we didn't see any defects in the amount of cells or the survival of the cells. So then we wanted to take this strategy that we had pioneered in HEC cells by transient overexpression and develop the actual lentivirus to deliver it to patient fibroblasts. But we started out with a control fibroblast line. And in this model system, what we had was control fibroblasts that express CT1 and presumably have a slightly higher level of creatine. And we delivered to them both AGAT and GAMT in separate lentiviral particles and allowed them to co-express both enzymes. And then we were going to do a number of readouts for how much creatine we could generate inside of a fibroblast. And similarly, with the patient fibroblasts, we took 2 fibroblast lines that had a complete loss of function in SLC6A8, encoding the CT1 transporter. And we also delivered both of these lentiviral constructs to those cells as well. So in wild-type fibroblasts, similar to HEC cells, we were able to get good infection, and the infection didn't seem to disrupt the amount of GAPDH when we load an equal amount of cells that were in the starting well, and we could get good expression of both AGAT and GAMT And in fibroblasts, there was definitely more GAMT expression when you check by Western blot for that V5 tag, suggesting that like AGAT might be not quite as abundant as GAMT, but the levels of overexpression compared to the endogenous amount were enormous. And so when we quantified the amount of intracellular creatine inside these healthy wild-type fibroblasts, we found a 12-fold increase in the amount of intracellular creatine. This was done in creatine-free media, but there's a little bit of probably creatine from the media that was exchanged to culture them. And so when we do this, the extracellular concentration of creatine in the media is also significantly higher. And we think that this is probably due to leakage out of the cells that are producing creatine on their own, because there's no exogenous creatine inside the media. And we use dialyzed FBS. And so then we also measured their total protein content, which was not different between cells that were infected with empty vector alone viruses or viruses delivering AGAT plus GAMT. And when you look by immunofluorescence, we wanted to see if these proteins are targeting properly within the cell. So in that paper from my postdoc, I showed that, that AGAT targets to the mitochondria and co-localizes with mitochondrial markers. And that's what we saw here, was that AGAT expression inside of these fibroblasts was in these sort of linear structures that were distributed throughout the cell cytoplasm, reminiscent of mitochondria, whereas GAMT was distributed throughout the cytoplasm and a little bit inside the nucleus. And I apologize for the lighting in here, but this is the same cells in 3 dimension. So we're just taking a confocal Z-stack and then turning it for you. And so you can see that the AGAT is localized properly throughout the cytoplasm. And we think probably the reason we get less AGAT expression, even though the viruses are titered to the same quantity, is because the AGAT distribution is so much smaller than the GAMT distribution. The mitochondrial volume is so much smaller than the cytoplasmic volume. And so what about in CTD patient fibroblasts? So the same experiment here, just measuring for protein expression. It was no problem to deliver these lentiviruses to CTD patient fibroblasts. This was the first line that we used, which had a deletion of exons 10 and 11. And when we did this, we were able to get good expression of AGAT and GAMT, and there's essentially no endogenous expression in these lines, and a similar distribution of the relative abundance of these 2 proteins. And of course, their expression looked very similar to that of wild-type fibroblasts. So it's the AGAT localized throughout the mitochondrial network, and the GAMT localized throughout the cytoplasm and a little bit inside the nucleus. The same cells in 3 dimensions. And then when we measured the creatine inside the cell, so I should say that each one of these experiments takes a month to grow enough fibroblasts to measure with the assay that we use from Sigma. And it just barely didn't reach statistical significance in this line, but I'll show you data from the other line. We were able to get about a 4-fold increase of creatine. And it was not significant in the media, and there was no effect on total cellular protein. But we tried this with another line of CTD patient fibroblasts, which had a different mutation. That is this W588X. And so when we do that, we get that same increase that we saw in wild-type fibroblasts, so like a 12-fold increase in intracellular creatine. And it was statistically significant in the media, which we attribute to probably a leach out of creatine from the cells to the media, since there's no exogenous creatine added in the media. And then the total protein content was not affected. So this is what the appearance of those fibroblasts look like. So we got, again, good expression, good localization, and a boost of 12-fold in creatine. So then we wanted to know, does this affect, so there are some salient features that we observed, like the total loading of our protein loading controls on the Western blot and the total protein inside the cell, but we wanted to see if this affected their cellular growth. And so what we did is we used this platform called an IncuSite, which is just a fancy microscope with a heating box on it. And so it heats the cells up like a normal tissue culture incubator, but it uses a phase microscope to watch the cells longer than you have the patients to do. And so it watches them over 72 hours and measures the percentage of confluence as the cells are growing out in culture. And so when we do this, when we overexpress AGAT plus GAMT, or we deliver empty vector constructs to wild-type fibroblasts, there was no defect in the cellular growth, so they grew indistinguishable from the wild-type controls. But when we did this with our CTD patient fibroblasts, so the CTD patient fibroblasts with AGAT plus GAMT are shown in red versus the empty vector control. In both cases, they were statistically outgrowing the empty vector control. And what I'll say about this is that we were just happy that they didn't die. So because overexpression of AGAT plus GAMT at this level, such that you're boosting the creatine content by 12-fold, could be potentially very deleterious because you would be consuming the precursors that are required to synthesize creatine. So that was good. And so the conclusions from this work is that lentiviral vectors permit high levels of AGAT and GAMT expression in HEC293 T cells and fibroblasts. Overexpression of AGAT plus GAMT increases intracellular creatine in wild-type and patient fibroblasts by up to 12-fold. The intracellular creatine content of CTD patient fibroblasts expressing AGAT plus GAMT is higher than the native amount of creatine inside of a wild-type fibroblast, so by about like 8 to 10-fold. So not only can you increase the creatine inside of a CTD patient fibroblast, but you can do so in a way overabundance of a wild-type in a natural level. And then this overexpression does not have apparent deleterious effects on cellular protein concentration or the cell's growth characteristics. So with that, I would like to acknowledge the people who helped to do this work. So through the funding from ACD, we were able to hire two technologists, so Chloe Wells and John Sorgenfrey, and they did all of the work here. So John headed up the project with HEC cells, and Chloe did the work mostly in fibroblasts. And they are incredible technicians. And I also should thank the folks the other folks in my lab. We work very closely with the Rudder Lab at the University of Utah, and then folks who helped me during my postdoc, and then other folks at the University of Utah. So with that, I'll take any questions, and I appreciate your time.
All right, thank you, Dr. Baker, for that. I guess we'll start with questions here. I do have one for you in a second, though.
Thanks, Steve. That was very interesting. I have a question for you. It's along the lines of what I asked Nicola at the beginning about the oligodendrocyte dysfunction. So previously, I've looked up in mice on a database for RNA-seq, and SLC6A8 is very highly expressed in oligodendrocyte and oligodendrocyte progenitors and myelinating oligodendrocytes. So I'm curious on your thoughts on this, because I asked Nicola, and he said, it's just very uncommon that we see myelination abnormalities in patients with GAMT deficiency or SLC6A8 transporter deficiency. But then you proposed possibly there's an unknown transporter, I think. Yeah. Can you tell us a little more about that thought?
Yeah, so the expression is heterogeneous, I would say, in oligos. So if you look at the single cell data, absolutely, yeah. So the expression of 6A8 throughout the brain is very heterogeneous. I wish I could show it, but even within neurons, it's not expressed in every single neuron on block, right? And it's just like that in oligodendrocytes. And I wonder if there is some sort of cell type specification amongst oligodendrocytes, like maybe all oligodendrocytes are not made equal. And I'll show you why. And if you stain for AGAT and GAMT, in the vita arbor, right, of the cerebellum. So this is going back to my postdoc now. So here, essentially all of these are OLIG2 positive, right? But there are some that have very low expression of GAMT and some that have massive amounts of GAMT. And this antibody is the same antibody I used on the fibroblasts. It's very specific. And so, and the same was true for AGAT. It's heterogeneous. And I wonder if there is some difference between oligos that actually need creatine and therefore would rely on it for function and survival. But there are other oligos who are dedicated producers of creatine and export it to the cells around them. And in that way, I would imagine that those cells do not express 6A8. And the reason why I say that is because otherwise you would set up a futile cycle, right? So 6A8 is a secondary active transporter, right? It co-transports two sodiums and a chloride along with each neutral creatine molecule. And so in order to perform that task, it relies on the energy produced by the sodium ATPase to export sodium, right? And so if there was a leak channel that allowed creatine to leave the cell and you express CT1 on the same face of the cell membrane, you would literally run a futile cycle. And so I'm guessing that there are, because of that heterogeneity in expression of 6A8 at the single cell level, that there are certain cells whose job it is to export, and then there are others who may actually use the creatine that they synthesize and actually uptake it from the CSF as well. But yeah.
Steve, question. Have you looked for any regulatory elements and their differences perhaps in these oligodendrocytes? This goes back to maybe work that was done by Schulze and Alec. Okay, in that there's a creatine binding element in the promoter region that might potentially regulate, synthesis of additional creatine. So so could that be, the differential maybe regulation of those elements be the reason why you have really high expression in some oligodendrocytes, less than others?
Absolutely.
And what would be a way to probably study that?
Yeah, and so it would be data like this. So I have to thank Brian Corses, who contributed this data, and I think he's now at UCSF. But when I was a postdoc, we worked together in the lab, and he did single-cell ATAC-seq to look at the chromatin profile of the gene AGAT and GAMT in human brain. So this is data from human single-cell ATAC-seq, which measures the DNA accessibility within the genome. And so if you look at the AGAT and GAMT loci, this is pseudo-bulk, so I don't want to get too in the weeds, but there could be heterogeneity here that is not revealed when you cluster all oligodendrocytes together. But suffice it to say that within the oligo lineage and the OPC lineage, particularly with feedback inhibition of something like expression of GAMT, so definitely there's this giant peak here, which is in the second intron of GAMT, which encodes AGAT. And that is accessible only in the oligo and OPC lineage, a little bit in astrocytes, but is not accessible in neurons and microglia or other cell types. And so you could imagine there are DNA regulatory elements in this stretch of DNA that are controlling the expression of these enzymes. And similar to the expression of GAMT, or GATM, which encodes AGAT, there's also GAMT, and GAMT has this large area of exposed chromatin in oligodendrocytes, which is not found as nearly as high in other cell types. And so what you could do is, using the single-cell ATAC data, is you could look and see if there's a correlation between, for example, the accessibility of SLC6A8 and therefore imply its expression with the accessibility of certain elements that are regulating expression of AGAT or GAMT. Yeah, to explain that heterogeneity at the expression of these synthetic enzymes and how does it correlate with the expression of the transporter. Yeah, that's a great idea.
So as I love the results, are you getting the same results on different variants such as a stop codon and the deletions?
Yeah. So the exonic deletion and the one with the truncating, both predicted to cause a truncation, but the exonic deletion deletes 2 exons, and so a large portion of the protein. And I think the exons are would cause a frame shift, and therefore no mRNA production. Production, but then degradation. But then the stop codon is an early stop codon. That would also truncate the protein during one of its final transmembrane helices. So both of those would be predicted to produce no functional 6A8, CT1. So we didn't check other fibroblast cell lines that have point mutations or that mutations that are specific for certain patients. But that being said, I suspect that since the function of CT1 is lost in all patients, at least partially, who have CTD, that our our mechanism, because it works in both wild-type fibroblasts and it works in 2 different mutant lines, would probably also work in a point mutant that had partial function of CT1, but we didn't test that explicitly. Yeah. So there is. So one of ours, the W, I think 558X, is a stop codon mutation. Yeah.
Hi, Steve.
Hey.
Wonderful data here.
Oh, thank you. Thanks, Jajji.
Yeah, we have done the similar experiments where we co-expressed AGAT and GAMT as well. Similar results. So, but your work is wonderful. And especially your figures of all this imaging, they're wonderful.
Oh, I spent my grad school in a dark room.
So where do you go from here? Like you have lenti AGAT and you have lenti GAMT, and you expressed in cells, they expressed it, made the creatine. Now what next?
Yeah, actually, that's a question I'd have for you too. So in essence, we proposed this as an alternative gene therapy strategy, as a backup strategy in case overexpression or delivery of 6A8 was toxic. And so we said, well, if delivery of SLC6A8 is potentially neurotoxic, perhaps we could try a different approach. And what really surprised me and inspired me, I would say, is your work and Troy's presentation last year at the Symposium, which really proved that you can increase the amount of creatine inside the brain with what appears to be no neurotoxicity. And I think Olivier Bressant also did not find any neurotoxicity in his model. And so, like, my thought on this is, yes, this works. But what it could potentially do to a neuron. So the deeper question is, why has evolution chosen the oligodendrocyte? If it wants creatine to be inside of a neuron, neurons only express GAMT at very low levels, but no AGAT. Why has it outsourced that task to an oligodendrocyte? There's probably some physiological reason why oligodendrocytes express such high levels of AGAT and GAMT, but neurons do not. And so, yes, you might be able to boost creatine levels inside of neurons by co-expressing AGAT and GAMT, but it may, in that context, be deleterious for that cell. It could be better than having no creatine, but it could still have drawbacks. And so, to me, the more physiological way of performing gene therapy is to replace the gene that's missing. And we pioneered this project because I was afraid that might be neurotoxic, but I think your work has proven that may not be the case. And so I would propose that they follow your strategy. Yeah.
I just want to say that sometimes evolution is not only that there is an advantage. Sometimes it's a symbiotic relationship, sometimes it's a parasitic relationship. So the oligodendrocytes were capable, and they had these 2 genes, and suddenly they said, let me wrap around neuron to help it. Similarly, we wrapped around mitochondria, engulfed it, and it was
yeah.
The cell became...
When we ate an alpha proteobacter, and that's why we're all here. That's right.
So it's not only always... true that, this has advantage, that's why this happened.
Yeah. Who knows if the oligodendrocyte had a choice here. Yeah.
Hi, thanks for the talk. I was wondering, why do you think there is such a difference between those 2 cell lines?
Between the 2 cell lines?
Yeah, the fibroblasts.
So I'm not sure. And what I would say is that there was one bad replicate in particular for that first cell line, which is why I think we didn't see as big of an effect. So, like, that, where did we go? Oh. The expression levels and things looked very similar, but like this one bad replicate, I think was, I'm not sure if somehow the cells got contaminated or something like that, but suffice it to say, since we saw the result in the other cell line, we were sufficiently happy, and it replicated what we saw in wild-type fibroblasts. And the other thing I'll say is that each one of these dots is a month of time. so the way we ended up in very early pilot experiments that Sangeeta and Heidi and everyone met with us, and when we were developing this strategy, we wanted to use the Sigma kit because we could do it in-house and we didn't have to wait for mass spec time. But the Sigma kit, which measures creatine, requires quite a bit of creatine in order to get a signal. And so the amount of creatine inside of a fibroblast is somewhere on the order of like a 1 to 2 micromolar, which is not so you need a lot of starting material in order to be in the linear range of that assay. So in order to do this, we called these experiments World War Z. So I don't know if you saw that movie, but with Brad Pitt, and I think he's inside the city, and all the zombies are stacking on top of each other to get over the wall. And so these fibroblasts, we would grow them up in these massive 15-centimeter culture dishes and let them become like 3,000% confluent to get enough cellular material to measure creatine reproducibly. And so in order to do that, it took 1 month to grow those fibroblasts to measure that creatine. And so we decided like not to waste another 4 or 5 months to add additional data points to the other fibroblast line. But yeah, that's a good question. Yeah.
I think we've got one more over here. Well, do we have time to run behind? Okay, sorry.
I was just curious because our studies kind of had the same hurdle of growing the fibroblasts to an acceptable amount, and so why choose this kit and say not like a method like LCMS for more sensitive detection?
Mostly because it was a plate reader assay and we had a plate reader, and it's a commercially available kit. And when we started it with hex cells, it works great. So there's enough creatine inside of a hex cell and presumably in a neuron, right? To measure with the Sigma kit. And then when we wanted to try this first in fibroblasts before we took it to neurons, we quickly realized that there's not very much creatine inside of a fibroblast. And so we were kind of stuck with the kit, but if we grew them for a month, then we could get enough material. And it's nice to be able to do something in your own hand, and there's always a wait for the mass spec at our facility. And so sometimes that wait can be months anyways. And yeah.
Okay, so this may be a help to both of you then. Just in the past couple days, I actually ran another available kit to all of us that ACD told us about, and we did this in nanomolar detection ranges for creatine uptake.
Nice.
So this may solve your time of growth, and we can discuss
Who provides the kit?
We got that from ABCAM, and you can do colorimetric for for micromolar, or fluorometric for the nanomolar. So if your plate reader does
nice. Yeah.
Fluorometric,
yeah.
Then that's what I went with because it is an increased sensitivity giving you nanomolar. So I could get this 4 days from growth of HEK. There are HEKs, granted, but from transfection to actual uptake was about 3 to 4 days for detection.
That's very nice. Yeah, that's useful. And hopefully as more and more folks work on creatine, the availability of these reagents will become better available and more known to us. So yeah.
So that was. No, I don't actually do the purchasing on it, so I'm not quite sure about that, but my boss got it for me and it worked.
Yeah, that's awesome.
I did have a question. So it seemed that this isn't necessarily like enzyme kinetics, but almost in a little bit that AGAT is almost like a rate limiting step where you notice that it was taken up in far less quantities, even though you were administering it in equal, quantities in your vectors, right? And so have you tried? And even in some of the Westerns, it looked like AGAT is a little less pronounced than the GAMT. So
so we tagged it with V5 so that you could use the same antibody to see both because of that reason. And we wanted to see what is the relative expression. Even though we titered these viruses to exactly the same titer, infected with exactly the same titer of virus, you get just way less AGAT out. And we think it's the volume of distribution. But yeah.
So have you tried obviously the work that you've done is great in a short amount of time, but have you tried dropping then the GAMT to kind of the level that you're seeing of the AGAT to match? Like if that's your rate limit, if you actually just need less and would still get the same outputs.
We probably can. Yeah, I think you probably could decrease GAMT expression and what the optimal relative expression of those two are for generating creatine, we don't know. And so that is a good question. Would it help to boost AGAT more? I think there is the danger of having AGAT without GAMT. And I think that was shown by Jerry last year, that you generate these other guanidino compounds that could be potentially toxic. And so it's almost a safety net to have a lot of GAMT around. And but yeah, what is the optimal concentration of these two enzymes is something to explore. Yeah, definitely.
Great. Okay, and so I think, do we have one more question? All right, we'll go one more, and then after this, we have time for a short coffee break before our next session.
So great talk, great idea, love it all.
Thanks, Matt.
Just a real quick question, or actually more of a spitball. The oligodendrocyte, creatine, AGAT, GAMT thing, could it be involved in arginine metabolism just in general from like glutamate scavenging, where you have these multiple pathways where you break down glutamate and you make arginine and you have to do something with that arginine and you don't necessarily want urea, but you need something. So we make creatine instead and more of a neuroprotective that way instead of maybe supplying the neurons directly with creatine.
Yeah, and then and you did see a little bit of expression in astrocytes, right, of like AGAT, right? And so that might be what those cells are doing with that, is not for necessarily production like the way that oligos do, we suspect, just because at a molecule per molecule level, oligodendrocytes actually express more AGAT than cells of the PCT. Right, so they're more able to produce guanidino acetate than the kidney, and so, but yeah, astrocytes also express a little bit of AGAT, and their job is to clear glutamate, right?
Right.
That's
Most of the support cells are in that sort of function as well, you know.
Yeah, no, and that could be a reason where you do see a discordance or something like that.
Just complete spitballing, just a guess.
That's neat. Yeah, it's a good idea. Yeah.
All right, so with that, our next coffee break is until about 2:45, and that is when we are scheduled to start session 3. So
Great.
thank you.
Thank you.